right, students, this is a short recording for Biology 1407 lab, and we're taking a look at the statistics assignment. And so we're going to go ahead and take a look at this a little more carefully. This is available in your Blackboard page. If you take a look at the third lesson in Biology 1407, and you go down towards the bottom, there's a statistics set right here. This is the one. Some students have been writing asking, where is this? How do I get it? Well, here it is. All right. And so we need you to answer each of these questions right here. The simplest thing would be to go ahead and print out this document and just fill it in directly. And so it has a separate, a series of questions in it, or problems, I should say, each of which has some questions in it. So here's question number 1, involving a distribution of cards, and you're going to use this data plus the chi-squared calculation. Here's the table right here. To answer the question, there is no difference between this distribution of cards and a random distribution, which should be ideally 400 each in each way, but you see it's a little bit different. That's the way statistics works, right? The more times you randomly do something, the more likely you are to get the true distribution that would occur if that thing is random. A small sample, in this case 1600, is not perfectly large enough to get a perfect distribution, but if you went to infinity, you'd get exactly 1/4 of each of the types of cards right here. That's the idea, and that's kind of an important idea of statistics. So the null hypothesis, null just means no effect, so there's no difference between these numbers and the numbers you're comparing them to, which is 400 versus 400 versus 400 versus 400. And an alternative hypothesis is the question we're interested in: does the poker card dealing machine have some sort of internal bias? And that's what we'll answer with the statistical test, and that's kind of the point of statistics. So we've got the rest of these problems in here. Each one deals with a variation on the same sort of idea, and they're all chi-squared questions, and we ask you to calculate them in here. They're not terribly hard. It should not take you a terribly long time. Students typically get very confused with this over here. We've got a number of different animals. We've got a group of 10 animals, another group of 10 animals we've used over and over for a total test of 100 animals. And collectively, we ended up with 35 of these animals that preferred a light environment in this particular situation, and 65 that preferred a dark environment in this situation. These are the numbers we're testing. This is the raw data that you would collect and put together. All right, and so on and so forth. So work your way through these. If you do have questions, come and ask me. Here is the chi-squared critical values table. And if you've been diligent with the lesson, you know how to work with this. But so we're interested in a 95% confidence that there is that the results of our any differences that we find in an experiment are due to the question we're asking and the particular intervention, the test that we're doing. We don't want the likelihood of random error to be too large. So you take a 5% random error, that's the one that you accept as being the normally acceptable one right here, and that's this right here. If you don't recognize it, 0.05 is the same thing as 5%. 0.5 is the same thing as 50%, 1.0 is the same thing as 100%. So this is a 5% allowable error in this column right here. And so the chi-squared value, 3.8, is what's called the critical value. And you compare the number that you generate using this table right here, which creates a chi-squared value. And if the value you create right here is larger than this number, what that means is the two things you're comparing are different from each other. In this particular case right here, it had to do with students in a Biology 1407 class. It has to do with students working with the ALU insertion right here. And we've got these distributions right here. You're going to calculate your Hardy-Weinberg expected numbers. That's this right here. And then what you want to do in the chi-squared test is compare the numbers we actually got in lab to the expected distribution. So here's the observed, and we'll put our expected right here. And we want to know, are those two sets of numbers different from each other? If they are, you're going to get a big value right here, which is greater than 3.8, in which case you'll say, yes, the two sets of numbers are different from each other. That's the way this works. All right. How do I know it's 3.8 and not 5.9? Well, that's the degrees of freedom over here. So if your problem only has two possibilities, three possibilities, which is what we've got right here. Homozygous for the ALU insertion, heterozygous, homozygous for no ALU insertion. Actually, the number we're going to be looking for, degrees of freedom, is the number of choices minus 1. So the number of choices is 3 minus 1 equals 5.99. So that's the number you're going to look at in this particular case. In this situation up here, looking at light and dark, these are the two choices. So you've got two choices. Degrees of freedom is the number of choices minus 1. So down here, that's where we'd use this 3.8 as our critical value. And so the random, you have to decide what the null hypothesis about animals' preference for light or dark, but I think that's fairly obvious. And then you're going to compare that expected distribution to the data you actually got right here. And if the chi-squared value that you calculate with this data is greater than 3.8, 3.841, then you say the numbers that we got in our experiment are different than the expected values. Okay? Up here, the very first question with the card shuffler, there are 1, 2, 3, 4 different possibilities of where the data will be. 4 choices. So you can figure out what the degrees of freedom is going to be. 4 choices minus 1. All right. That's a little bit about your homework assignment. Get that thing done as soon as you can. It should not take you long. And you're going to upload it right here in your statistics problem set. You're just going to upload it right here. Browse your computer for the finished document. However you want to generate that, you can take the actual document right here and fill it out. You can do it by hand. Best would be to print this thing out right here, fill it out, and hand that in. It makes it easier for me to grade. Making it easy for me to grade makes it easier to, grade. Big relief for me. It's due by Sunday night. Okay. Your ALU report. Right. That's also in your lesson. So let's go ahead and take a look down at the preparing your research paper in your lesson. All right, so here's the instructions for your research paper. You can print these out if you want as well, but this tells you what to do. I'll walk through it, okay? But there's the instructions. The big thing I want to tell you is, especially if you've got anxiety about writing lab reports, you're really not sure what the heck you're doing. If you weren't a student of Biology 1406 at Northeast Campus, where we make you write papers in the first semester, you might not be particularly used to this, so you have to get up to speed. That's okay. Get it done, even if it's a terrible paper, by the deadline. That way I can go over it using this grading tool right here. This is what I will hand back to you, or I'll email to you in y'all's case, because we're not meeting face-to-face. But this is what I'll use, so for, let me see if I can get up here and show you that. So for the title, which has to have a name and group number, the title in your name, is 3 points. The abstract, the purpose, the results, the conclusions, 2 points each, etc., etc., etc. All the way through here. Just do each of these things. Write your paper, write the question to be answered. Next sentences. What are transposons? What is an ALU insertion? How does the ALU insertion show up in the primate lineage, and what's the importance of that? Don't know anything about this? Why don't you read about it? Get on the internet. For our biology class, a good reference that'll be really, really handy and an easy place to look, always in the same thing, always in the same place for equivalent information is Wikipedia. That's perfectly great for this biology course right here. When I said get on the internet and look up the information, remember, you can always get bad information on the internet. What you're looking for is stuff where somebody's put their name next to what they've written, or in the case of Wikipedia, there's lots of references. Where did they get the information that's in the article you're reading? Those references are always at the bottom of the paper or the article in Wikipedia. Anyway, read about ALU and the primate lineage. Okay. And you work your way through here and get this thing in by the deadline. If the grade is bad, when I grade it using this thing right here, you'll have a chance to rewrite it. And that's the whole point. But it's impossible to rewrite it unless you write something and hand it in. So please go ahead and get this document done. Okay? And that, again, is due by Sunday night. And that's been on our schedule since the beginning. It's in your calendar that's built into the syllabus document that starts your semester. So, you know, use the tools that you got. And, yeah. All right. In Blackboard, when you want to upload the ALU report, go ahead and go. It's in here in your table of contents, the ALU report. Go ahead and click on that. Go in here. You're going to complete it. Please note these rules. The paper has got to be in a PDF or a doc or a docx or an RTF, something that a PC can read. If you're using a Mac, please remember to save specifically in one of these formats. You'll probably want to go for the PDF. If you haven't changed the settings on your Mac device, it might save the paper in a proprietary format that only another Mac can read, and TCC issues us PCs. That's what we work with. So please attend to that. It's due Sunday night, 11:59 p.m. And remember, it doesn't have to be perfect. It needs to be in. You can always fix it. Okay? And that'll be the tail end of the work on week 3. I've recorded and posted a video on interpreting your gels to write the report, so that'll help a little bit. Okay? And that is about it. Okay. Thanks.